in a line while you're interested in brain aging and longevity is because one of the primary factors, like as we age, of course, all the organs are aging, but somehow brain aging really can predict how long you're going to live, right? So you can probably fix all other organs to some degree, but yeah, brain aging is still a complex subject. So, and it is one of the most important, way of how you can, or factor which can determine your healthy aging, right? So I think that's why we want to cover brain aging and longevity today. So we have, apart from me, three other people here. Isaac John, who is founder of Ivory Tech. We have Professor Suvarna Alladi from NIMHANS and Professor Ravi Muddashetty from CBR. I'm Dr. Shovamayee Maharana from IISc. I'll first introduce myself and I'll then ask each one of you to introduce yourself in a line or two or in a minute or so that we can quickly go to the questions. So I am working in ISC from last 4.5 years. What we do is to understand how do neurons die because of protein aggregation, which often happens as part of neurodegeneration condition like ALS or FTD. So ALS is amyotrophic lateral sclerosis or frontotemporal dementia. This kind of aggregation is also common in some other rare neurodegeneration. So we do look at how proteins behave in neurons and then study these disorders. Maybe I'll now ask Professor Ravi, since he is also a cell biologist like me and a basic researcher. Yeah.
Hi, everybody. I'm Ravi Muddashetty. I'm a scientist working at the Center for Brain Research, which is in IISc. So my primary focus is on memory, particularly on the cellular and molecular aspect of memory. I started as a scientist working on neurodevelopment, but more from 10 years, I've been also working on neurodegeneration, and I primarily focus on synapse, the healthy aspects of the synapse, physiological aspects of synapse, and signaling, energy metabolism, and other aspects, and what goes wrong in this aspect, which could lead to the pathology, particularly the loss of memory, dementia, and neurodegeneration.
Yeah. Thanks, Professor Ravi. Professor Aparna, maybe I will ask you to Suvarna, sorry. Introduce yourself. Yeah.
I was looking for Aparna. So hi,
I should. My memory for names is not so good.
no, Suvarna and Aparna are close enough.
Yeah.
Yeah. So hi, thanks for having me on this webinar. I'm Suvarna Alladi. I'm a neurologist. I'm a cognitive neurologist, professor at the National Institute of Mental Health and Neurosciences. So I coordinate a large service for clinical service for people living with dementia, and we see people across the entire spectrum of dementia. The diseases that you heard Shova mention, frontotemporal dementia, ALS and dementia, Alzheimer's disease, vascular dementia, Lewy body dementia, all of which have protein and molecular correlates that people are working in basic science. We see the clinical manifestations. We try and have better ways of diagnosing them. The field is moving towards biomarker-based diagnosis. So far, it was just MRI, which doesn't give all that much information of what's actually what protein is aggregated in the brain. And then we also provide treatment, multidisciplinary care, caregiver support. And what is exciting now is we have new anti-amyloid therapies for the first time approved by the US FDA. So those monoclonal antibodies against Alzheimer's are coming. They're approved now in India. So we are in the process of developing appropriate use recommendations for who should receive, who should not receive. And so we're really prepared to take care of these people. But that's just still a very small proportion. So also look at the question that is asked to us in the clinic is how can we prevent dementia? What could I have done to prevent dementia in my father or mother? So we look at public health or epidemiological studies. We go into the community and look for risk factors for dementia, protective factors, and I'm part of this Lancet Commission, which has given epidemiological evidence that 45% of dementia risk can actually be identified. So the gamut really of prevention to diagnosis and care, and policy for advocating for people living with dementia in policy. So we have a brain health initiative that's running across the state and now across the country as well in a few districts. So everything but basic science.
No, I think it's really, really cool that we have this whole range. And now I think over to Isaac. Or how do we call you? We call you John or we call you Isaac?
No, Isaac. Isaac is good. That's my
Okay.
first name. Thank you for checking, show me. So pleasure to be here with such an esteemed group. My background has been in consumer businesses. My personal background, however, and the reason why I resonate with the subject a lot is because my father had Parkinson's. So I've seen that journey from, being a caregiver to also being someone who was very helpless, in managing a neurodegenerative condition. And that sort of spurred me and my co-founder who also had a grandmom who had dementia. So we were just like curious students in a way of the field. And as we came closer and closer to the subject, we realized that early cognitive screening is unfortunately, very, very, sparse in India. Hardly any health packages offer it. And our goal is to advocate for better brain health through both early cognitive screenings and relevant cognitive interventions.
Cool. Thanks a lot. So I think what I will do is to really start with some questions which were given by people who registered. And this is just to start a discussion. And I think we all can ask each other questions. And at the end, around like when we have 50 more minutes left, we will also get questions from the audience. But if there are any burning questions from the audience, please type in the chat. And if it is, going to be in the relevant direction of our discussion, I will pick it up and I'll give it to our panel for discussion. So I have my chat window open. Yeah. So, okay, I think let's start with Professor Suvarna, since she's the one who really sees, people who are suffering from, which we can say is like an advanced stage of brain aging related disorders, right? So, what do you think what you see in your patients? Are they all elderly, or do you also see that there are people who have, say, other chronic disorders also have brain aging related disorders?
So thanks, Shoma, for the question. Advancing age is a single most risk factor for dementia in general. So advancing age is not a cause, but it's a risk factor. So the older you are, the higher the probability of developing dementia and brain pathology that causes dementia. Having said that, when you talk about younger people, in the West, the mean age is around, between 70 and 80 or even above 80. Most of our memory clinic studies have shown that in our population, it's around 64, 65. So I don't, typically they say less than 65 is early onset dementia, but that's in the West. So definition of early onset is moot. But we around 64, 65 is when they come to us in the clinic. So the symptoms would have started around 60. And of course, we have people in their 70s, we have people in their 80s, we have people in their 90s also who develop dementia. And that is about typical Alzheimer's disease. But there are some young onset dementias which are degenerative in origin that typically affect people who are younger. For instance, the disease that you work with, frontotemporal dementia, is a young onset. It's a typical age is around 55, 60 that they start developing behavioral problems. It's often undiagnosed. Then you can have other diseases like progressive supranuclear palsies, another tauopathy. They can come early with dementia and extra Parkinsonism. Then there are some unusual metabolic diseases which can present in even in their 30s. So dementia is a disease when a person is born, develops normally, has had a adult life, cognitively fulfilling life, and then they start showing cognitive decline. So that can happen even in some inborn errors of metabolism Stroke is a cause of young onset dementia. Young strokes are very common. So if you have a stroke in the age of 40, it can damage the brain and you can have dementia. So vitamin deficiencies, autoimmune disorders, tumors. So there are so many other brain disorders can come very earlier than Alzheimer's disease. So it's not maybe about in our clinic about 20 to 25% of people are people who come in their 50s to early 60s, and the rest will be about 65 onwards. You are muted.
Yeah, so the around 25% of people will come at younger age
Yeah.
for dementia related things.
Yeah.
Okay, and as you said, metabolic disorder is part of that spectrum, or stroke or other things in younger population.
Right.
But then aging is the major factor, right? Um, but when the people come to you, are they already in an advanced stage where it is like, can there be still treatments? There was one question in the chat box saying, for example, does PSP has a treatment, or say Alzheimer's, or is it only
Okay.
about managing the symptoms?
Right. Till recently, we had only symptomatic treatment for dementia. But I think just about more than a year ago, the first drug for Alzheimer's disease was approved. Now, these are not cures, these are monoclonal antibodies that deplete the brain of the amyloid protein. So the amyloid levels in the brain come down. And what the trials have shown is that there's about 30% slowing of decline. So if you show the graph, the
I
people
see.
on the drug over 18 months, they slow much slower than, they decline much slower than, so they would reach from the early stage to a mild stage. They would take a little longer to reach the next stage. So, but these
I
are
see.
disease modifying treatments. And then have multidisciplinary care. There's a question I see here on social isolation,
Yes.
loneliness status. Of course, social isolation is an important risk factor in the Lancet Commission. But in COVID, we saw that not interacting with one another, our patients with dementia declined so fast.
Yeah.
So social engagement. So you can put in multidisciplinary care, cognitive stimulation therapy, social engagement, physical exercise, physiotherapy, activities, and helping them do activities. All of that also are part of the treatment. It's not just drugs. That's the
Correct.
point I'd like to make. Yeah.
Correct.
Environmental modification. Just by changing the lights and the color of your walls and removing some cluttered objects on the floor and making sure there is not too much distraction in the house, you can make a person better. Yeah.
That's very interesting. I think at this point, I'll also ask Isaac. So, Isaac, what is the goal of Ivory? Is it only on detection? Because that's what we were asking right now,
Right.
Suvarna, right? That when do patients come? Is it possible that this can be detected earlier? And then, how is your app or your company going to help? Yeah.
Sure. So at Ivory, we basically, like I said, the goal is to actually have early cognitive screenings plugged in as many, let's say, healthcare providers at place. And in this, I'm happy to share some data in this regard as well. See, in UK, in US, they actually have something known as an annual wellness visit, the AMV. It's actually funded by most insurance companies. And there, anybody above the age of 45 will actually get a compulsory cognitive screening done. And UK similarly has got certain packages. Singapore has these packages, in health screening packages. Whereas in India, if you look at the landscape of health checkups in India, so much of awareness about heart health, kidney health, liver health, but almost nothing related to the brain, which is the single largest predictor of like how great your health span can be. Right, your brain aging early is not a great sign. So our goal at a very high level is to actually be plugged in with as many healthcare providers as possible so that early cognitive screenings become like almost as mainstream as, let's say, your glucose blood monitor or any of the other tests in a non-invasive manner to begin with. If there are risks detected at stage, typically our channels are clinicians and hospitals wherein there are relevant clinicians in place to actually take
So,
for
Isaac,
next course of
how
action.
early the cognitive decline happens? Does it happen much before? Or maybe Suvarna can also answer. does it happen much before the brain has a phenotype, right? Like you are measuring cognitive decline, and then I think there would be changes structurally inside the cells and regions of brain. So how are those two related?
Yeah, I'll let Dr. Suvarna answer and then I'll follow up.
Yeah.
So the clinical phenotype always follows the brain changes. It's not the other way around.
Mm.
So what happens is first amyloid accumulates in the brain. Typically by 45, 50 it starts. And then there is a very nice, I mean, since there are students here, you can look for the Clifford Jack curves.
Mm.
And they'll give you a very nice delineation of when the amyloid starts accumulating. Then when the cerebrospinal fluid starts showing lowered amyloid, and when the tau levels in the CSF go up. All this is before clinical manifestations.
I see.
And then you have the tau in the brain accumulating. And that really signals the clinical symptoms. It's not really when the amyloid is accumulating. So tau accumulation is very mild. We call it, they're all pre-mild. So you have normal cognition, then you have pre-mild cognitive impairment, and then you have mild cognitive impairment is when tau starts accumulating. And all of these have a gap of about 5 years or 10 years between each other. So there's a lot of time in between to do something useful for your brain. And then once tau accumulates, and then the brain volume starts shrinking. And that is in the hippocampal volumes. When the hippocampal volume starts shrinking, what starts happening as degeneration sets in, there's an NFL, neurofilament light chain. And that level starts dropping. And it is when the volumes actually start showing decline, when people start showing symptoms of dementia. And that is even then, it is still mild dementia. And only then, yeah. So you can see there's a 10 to 20 year changes in the brain happening. That is in a garden variety sporadic. I'm not talking about genetic forms where it's very different. But yeah, so it's and I think at the cellular level also it's like that. The synaptic changes, all of that happens. First, the connectivity comes down. So I can ask basic biologist Dr. Ravi to maybe tell us a little bit about at the molecular level. I'd like to know. So this is our story, based on lots of imaging data and longitudinal studies that have been done. So what happens at the cellular level and then maybe Issac as well. So
I think Ravi.
Okay, thank you, Dr. Suvarna. So yes, so clearly the cellular and molecular pathology is the beginning of the whole disease development. So protein aggregation is one of the key hallmark of many of these kinds of dementia. So, I mean, as Dr. Suvarna mentioned and Shova, you also mentioned, there are many, many kinds of dementia. So, Alzheimer's disease and Parkinson are the major portion, but there are many, many different. But even in most of this, there is accumulation of protein aggregates is one of the key things. So there are two reasons for that. There could be genetic component which leads to the excessive protein fragmentation that leads to the aggregation. That is one of the which is classically studied in case of amyloid beta protein fragments. But in many other cases like synuclein or in many prion disease, there is no fragmentation. There's just the accumulation. So one of the reason is abnormal structure of the protein, which needs to be cleared. So there is a very active clearance mechanism in cells. And these cells in the somatic cells, like, if there's something goes wrong, the cells can be replaced. But in case of neuron, there is no replacement of cell. If any problem in the machinery, which has to clear this aggregated protein or excessive proteins, that leads to the slow accumulation of this protein. And this is a kind of vicious cycle. The more accumulation, more defect in the clearance mechanism. So there will be, but brain, as Professor Suvarna can elaborate, there is a lot of redundancy in brain and its connectivity. There's a huge amount of redundancy. This is actually a problem for the early detection of dementia. So because of the redundancy, those cellular and molecular phenotypes come very, very early. It can be pointed out if we had a way to look at the cell. And if we had a way to look at the molecular changes happening in the neuron, we could probably identify the defects maybe 20 years earlier before you could see the clinical manifestation. But unfortunately, we cannot. But when it comes to the phenotype of the memory, there is a huge redundancy in the brain, particularly in the cortex. Because of that, though the molecular and cellular defects keep accumulating, you will not notice the actual decline in memory and the cognition. So that is the reason. But the protein aggregation that leads to the synaptic defect. Synapse is the connection which is responsible for all our memory and cognition. So protein aggregates particularly affect the synaptic connections. Once the synaptic connection is affected, it affects the signaling, the physiological talking between the synapse. These are the things which are primarily affected. But as I just mentioned, it takes a very long time to actually from here to the cognitive decline.
And that's what Professor Suvarna was also saying, that the brain, even when the brain shrinks, you still have very mild cognitive disability, right? And so when does the so Ravi, in your studies, what do you see when you have cellular aggregates? Is it now a one-way street that now there is no way going back and cells are just going to die, or there are ways to still reverse it or rescue it? What do you think?
Yeah, so as Professor Suvarna already mentioned, there are two major kinds of aggregates. One is extracellular aggregates, another is intracellular aggregates. The extracellular aggregate is primarily amyloid beta is one of the most well studied. And there are many intracellular aggregates like tau and synuclein. So I can say that extracellular is probably much easier. I mean, there are whatever therapy so far has been tried and has been approved. They're all for the extracellular accumulation. So there is if like beta amyloid, even if there is accumulation, if you can use monoclonal antibody or any other way where you can remove, it's much easier, relatively much easier to remove the extracellular aggregate. Then the reversal is possible. Once you remove these aggregates, the cells can start behaving whatever but this has to happen before there is considerable amount of neuronal loss. As already Dr. Suvarna mentioned, neuronal loss happen much later. So if the removal of extracellular protein can happen before the loss of neuron, lot of reversal is possible. But if the neuron loss happens, then it's difficult. But the tougher problem is to target the intracellular protein aggregates. So intracellular, obviously, your antibodies cannot go inside the cell. That's much tougher. Then the only way is obviously the neurons and every cells have their own mechanism to clear these proteins. One of the most important mechanism is autophagy. So autophagy is a mechanism which recognize the non-functional proteins, cleave them and remove them. This also includes the lysosome. Autophagy along with the lysosome is a mechanism. And a defect in this mechanism is one of the primary cause for the cellular pathology. One way you can reverse is by activating this mechanism. You can activate autophagy, you can correct some of the lysosomal defect. These are still at the preclinical level. As far my knowledge goes, there is no mechanism or drugs which are still targeting this, but these are very promising. Particularly activating autophagy is a very promising area for therapy.
Yeah, I think activating autophagy in general might help also in other organ aging, right? As in, that's why people say, okay, do fasting, and maybe fasting can increase autophagy, and that can. I don't know how much data is out there. So, okay, I think Isaac, I had another question for you. So we were talking about this detection, and okay, so it looks like biomarkers accumulate 20 years in advance, right, before you actually have a cognitive decline. Or you think there are better cognitive tests which can be devised, which can even start detecting when we are in this biomarker accumulation phase, when we are in the tau accumulation in blood, or what is the status there about cognitive tests?
I think things are changing actually pretty fast in this world, wherein almost every cognitive assessment, while seems like it can be great at, let's say, only looking at, how executive function is impaired, or how, let's say, memory is impaired, or how, let's say, attention is impaired. So I think what's really encouraging to see in the assessments world and screenings world is that there are different companies across the world I'm seeing which are kind of becoming specialists. And there are specific tools which can be deployed both for general cognitive impairment as well as for specific conditions as well. So that's very promising to see. What is, however, very clear is that signs of cognitive impairment, when evidenced through, let's say, these signals and compared with large data sets over time, normed with large data sets over time, especially if they are culturally kind of specific and sensitive, then I think it can go a long way in catching those subtle signs. I
So you
don't
mean to say that you have to couple your cognitive test with
with
biomarkers,
yes,
also with your cultural
yes,
environment, to make it more sensitive, because currently it looks
correct,
noisy.
correct.
Yeah. Okay.
I don't think, I mean, I would hope if Dr. Suvarna agrees, but to just take only a cognitive assessment score and say that, so and so person is diagnosed with this or is definitely,
Is
I don't
not
think
enough.
our sciences
Hmm.
reads. Where
Hmm.
science has progressed considerably, kind of making more, condition specific kind of screenings is where our world is. So I was just coming to that, the previous point, and we always feel that a company like Ivory benefits when the tool is like matched with other tools that are available with, let's say, a clinician, right? So at a very high level, our goal is to be plugged in with the right relevant providers and points of care.
Yeah. But does this cognitive test will change? For example, has anyone looked at, for example, with aging, and as Suvarna was pointing out, that in India, the mean age of dementia is 60 to 65, whereas if you went into Western countries, it might be 70 or 10 years
Yeah.
ahead, right? So we probably age brain-wise faster than our. Is it true, Suvarna? Like Indians are aging, brain aging is faster than Western or
Yeah.
populations.
So, first thing first,
Mm,
our average lifespan in India is at least 10 years younger.
that is
So
true.
we
Yeah.
live much, we don't live as long as the average high income country person would live. So, but I
Mm,
think, so if you look at the curve of people in at different ages, we don't have too many in their 80s and all of that, right?
mm,
So it's just that there are more people in their 60s and 70s, and in that there's a proportion who develop dementia, and therefore our population has a higher proportion of younger people because we have many more younger older people than older people.
mm,
That is one first explanation before we, So then when you look at, I know, Parkinson's disease, or if you take stroke, or you take Alzheimer's disease, there is consistently that if you take age adjusted prevalence rates and you adjust for the age, it appears that they they do come a decade younger, right?
mm.
And then there is emerging evidence about something that people are calling accelerated brain aging. This work has come from many groups, but Latin American Augustin Ibanez's group recently has published a very nice article in Nature or somewhere. They
Mm,
talk about social determinants of health,
mm.
and so which impact brain aging, and that if you take social determinants like economic status, vascular risk factors, stroke, poverty, stress, pollution, all of the
Yeah.
other things that are so
Mm.
different across high and middle income and low and middle income countries. So these are factors that might uh, contribute to accelerated brain aging. There are lots of questions I saw about how the molecular
Yes.
mechanism is.
Mm.
I think the basic biologist should tell us, but there is a general trend emerging that maybe there is some accelerated brain aging in our population, but that in India, I don't think we have demonstrated it at all yet. That's something that
Mm.
needs to be done. So for that, you need to do longitudinal MRI scans, longitudinal cognitive assessments, and you really need to have a robust group data to demonstrate this. In India,
Also,
we
I
don't
think
have the
in the
data.
way we are also unable to follow our patients continuously, right? I think that's one thing which lacks in India. For example, we don't have a very strong unified insurance system where we can track people, right? I think that you might have come across this as a hurdle to do longitudinal studies, right? Or
Absolutely. I think that research, well-defined longitudinal studies, I know CVR is doing some, we are doing several cohorts. Lassie Dad, longitudinal study aging of India is a huge study that's looked at repeated MRIs. They have, I think, a cohort about 4,000 that they've actually done the repeat MRIs.
the CVR is the pioneer in this, I think, in the country
Yeah.
where they really
So
following urban, rural also in context of that. Yeah.
these cohorts will tell us about brain aging eventually. In our context, there are all these cohorts across the country also. So they will tell us about accelerated brain aging, and its attrition is a problem. People do not, they're not research literate in that sense. So unless we, and the other thing is, I mean, you do all the tests, but if you're not providing care, in the West, there's a clear care pathway and there's a research pathway, and then you don't do care when you're doing research. And that's something that's a bit challenging for us, because the person who you come home and you do all the tests and then you find a problem and then what, right? So I think we have a lot of work to do to improve our, and even our dementia patients don't come back quite often to us. We have a very sharp decline in follow-ups for many reasons. So you're right, follow-up in India is a challenge, but I know CBR is doing a good job. Their attrition rates are much less.
I think if more people are aware that everyone can contribute to this research just by becoming part of these studies and just going to follow up in the studies, and I think all of us can contribute, and then I think once we have awareness, we'll have much larger cohorts. So I think I want to answer one thing, because we have seen in our in vitro studies that looks like oxidative stress is one of the most important thing which leads to aggregation of these proteins, and you can get oxidation stress in various in various ways. I eat more calories, or I'm stressed, or I live in a pollution environment. All of this feeds into your body and then leads to oxidative stress in different ways. That's why eating healthy, which means a lot of antioxidants and vegetables, fruits, I don't know, kind of hand wavy right now, but looks like oxidative stress at cellular level really leads to aggregation. I don't know, Ravi, you have some studies like this in animals where you have seen it is true, because we look at in cells. I don't know, are there animal studies which also says that oxidative stress is the main stress to drive all this, or?
Yes, oxidative stress is one of the very key cellular pathway which leads to the I mean, of course, you can have already the mutations like as I mentioned, APP7 mutation, we or even alpha synuclein. The mutation can also lead to the initial aggregation, and but as you rightly pointed out, oxidative stress, particularly generating lot of reactive oxygen species, lead to further activation of this protein and leads to the aggregation. And this make it much tougher for the cellular clearing mechanism to get rid of. Not only that, once you have the increased oxidative stress, it affects all other signaling pathways. So once the signaling, particularly it affects your mitochondria, though the mitochondria is the primary source of reactive oxygen species, it also the primary affected organelle. Once the mitochondria get affected, your overall the ATP synthesis, energy synthesis goes down. To push it further, the mitochondria will do overwork, which will only increase the oxidative stress further. That will also lead to the endoplasmic reticulum stress, and Golgi get affected. So basically, it starts with mitochondria, spread to all the organelle. And as I said, I mean, of course, it affects all kind of aging, but particularly in brain. So it affects the synapses and the connections, which finally leads to your loss of plasticity.
Yeah. So there was this question somebody has asked about, does aging affect organelle dynamics, transport, and positioning in neurons, which leads to neuronal dysfunction? So I think we discussed it in context of oxidative stress, but even without oxidative stress, with aging, does it also happen? Yeah.
So one of the things, particularly if I can talk about mitochondria, in case of neurons. So mitochondria is very heterogeneous. We somehow in the school, whatever we study is it's like, it's just as one bulb kind of structure which makes ATP, but it's a very heterogeneous. So it has very small mitochondria. Some of the places it has very long branch mitochondria and so on. This is particularly important in case of neuron, because in the neuron, if you look at the cell body, mitochondria size is smaller. But as you go further into the dendrite and axon, further away from the cell body, mitochondrial length becomes bigger and bigger and much branched. So these are more stagnant mitochondria compared to the very dynamically moving mitochondria in the cell body are close to the cell body. So it is very important this mitochondria to be transported to the dendrite because it's more stagnant. If some part of the mitochondria is defective, that has to be removed and has to be replaced. So this rejuvenation is very required. So as we age, the transport and also the as I said, the mitochondria keep splitting and fusing. This fission and fusion is a very active process. So as the aging increases, the fusion goes down and fission increases, and this is problematic. So you don't
Also,
have this
amyloid,
long.
will the amyloids change this transport? Like if you had amyloids in your neurons, intracellular amyloids at say the axons and so on, then this transport might also get blocked, right? And
Well, I think the tau may have a major role in the transport than the amyloid, as we already discussed. Amyloid, I mean, of course, amyloid also get taken into the cells, but a lot of the aggregation of amyloid actually happens outside the cell. But it's the tau and synuclein. These are the ones which are get accumulated inside. Tau directly has a role in the transport, microtubule based transport. Most of this organelle transport happen on this super highway called microtubule in the cells. And this is all the more important in case of neuron because this neuron is a highly structured thing. It is very so you have to send things to the required place. If it doesn't happen, then these are highly decentralized. It's unlike the other like cells. Neuron is highly decentralized. The functioning of the synapse is locally regulated. Everything has to be supplied there. If the supply chain is defective, of course, you will have the consequences very clearly in neuron more than other things.
more than other thing. Yeah.
Yeah.
So I see this another question. Is there a difference between brain aging between men and women? I think Isaac, you want to say something? And then I can ask this to Professor Suvarna as well.
I don't know it
So
from a clinical. Sorry, please go ahead.
please go ahead. Please start.
No, I was just going to say that I think I'm not equipped enough to answer this question, but I do know that women by default are given their longer lifespan as well as the hormonal changes that go through are of course at greater risk of Alzheimer's and dementia. But whether that translates to how does that translate to? I think it will be better.
So, Suvarna, Professor Suvarna, do you also see a kind of bias in your patients where you see between men and women, they age differently for brain health? And
yeah. So, like Isaac said, women are indeed more vulnerable to the development of dementia. So that is true. And we don't, and then, but brain aging studies have shown that the biological age in women, brain age in women is more commensurate with their biological age. But in men, maybe it's a little more. So these studies are not confirmatory at this point. But what we know is that they are more vulnerable. What we definitely know is that the perimenopausal state and the lack of estrogen and progesterone is actually accelerates development of brain disease. We also know that the healthcare parameters and health-seeking behavior for women is much less.
that is true. Yeah.
So I think the more the depression is more of, so the more proximal at not really so much at the biological level, but more at social determinants of health level. Women are much more, they will come late to the hospitals. They will get much less care. They will progress faster. They have more depression. They have higher untreated diabetes and hypertension, obesity, inflammation, all of those other modifiable risk
Chronic conditions
factors.
are also
Are less attended to. I
are
won't
less.
say less attended to and less treated. And
Correct.
of course, menopause is a major big risk factor. So I think this
Yeah.
much we can say. And we also know that they're prevalent, that they are more vulnerable to developing Alzheimer's disease. Yes.
Yeah. So there is one question in the chat box which says, okay, if you have detected, screened or diagnosed for neurodegenerative disorder, what will be the next step? Like, what does happen? Can we control it? Like, what will you change in patient? The diet, the lifestyle, or will you give them some medicine? So I think there is a general question like that in the list of questions I have. Yeah.
Yeah. That's what we do, right? My job is
But
exists because
overall, what does
we
major
diagnose.
change?
What I'm trying to say is,
Hmm.
after diagnosis, the work actually starts.
Yeah.
And it's also, you have to diagnose what degenerative disease is it, which kind, what stage at which it is in, and then what are the, of course, pharmacological treatment, and then, like I mentioned, there's a lot of rehabilitation that can be done. As I mentioned, caregiver support. So there's a lot of care of the family and the patient that starts there.
I see.
It starts there, and so we need to engage constantly with the family, because every stage of the disease brings its own unique problems. It's not like you make a diagnosis and it's over. It really, the engagement starts there.
Yeah.
And there are such few specialists, such few multidisciplinary care teams. I think everybody here will agree, if you've known somebody with a neurodegenerative disease, whatever you do is not enough. Whatever you do is it is so difficult, because it affects behavior, it
Yeah.
affects movement, and it affects cognition. So it's
Correct.
like a triple whammy. So
They
you
need a lot of support and
need a lot of care and treatment, proper treatment,
and
and
treatment.
identification of problems. They're confused. They are
Hmm.
not refusing to eat. They are not able to swallow. They are getting aggressive. They refuse to get up and have a bath. So each of these is a symptom that requires very specialized care. There are speech therapists who are specialized in this. So the teams, physiotherapy, they won't do physiotherapy. So you need a physiotherapist who understands that a person will forget the exercise as soon as you tell it to them. And how do you make them do it? It requires specialized skills to manage a patient. So you need dementia, degenerative disease, specialized teams, which we have in NIMHANS, but we literally reach a very small proportion of people. So there
Yeah.
are a few groups that are trying to do this, but it is nowhere near enough. Nobody is satisfied. We are not
Yeah.
satisfied. Patients are not satisfied. So you have new startups. Therefore, everybody is trying to make it better, but it's
Yeah.
very hard. It's very hard.
So I think, yeah, what you're saying is we really need a systematic approach to identify people, to support them. And I think, yeah, with your connection with government policies and so on, I think you guys are trying to make a structure, right, to these kind of support services. But
We've
what would
developed.
be,
Yeah.
yeah, yeah, please go on, Suvarna.
No. We for the only state in India. Am I echoing? Echoing?
I think
No,
it's
now it's
now
better.
better.
Yeah.
Yeah.
Where we set up a brain health clinic in each district hospital.
Yeah.
And there you have 6-member brain health team. So you have a physiotherapist, speech therapist, psychologist, a physician, a nurse, and a district coordinator. But the
Yeah.
numbers of people seen there are very few. Awareness is very low. But at least the government is funding this, and we are training them, and we hope over the next years we will actually make a difference. Yeah.
Yeah. So I think before I take all other questions from the box, I have one last question for all of you. What do you think? So basically, it looks like prevention is better here, right? In case of brain aging, what is the most important thing we can do in our, say, lifestyle, which will make a big difference for brain health? Maybe we start with Professor Suvarna, then go to Ravi and Isaac. Yeah.
Isaac is the only one standing as we are all sitting.
Oh, that's the best thing we should do, I guess.
Absolutely. There were lots of questions on physical exercise and brain health. I think um, uh, physical exercise, controlling your body mass index, reducing insulin resistance, controlling vascular risk factors if you're hypertensive, diabetic, alcohol, smoking, absolutely. These are the 4 things that we can, and substance abuse, the first 4, 5 things we can immediately, tackle, and they're all difficult, yeah. Then reduce stress.
Meditation. So somebody asked if meditation is effective. I think if it reduces stress, then maybe, yeah.
Yeah, but it's so difficult. No, have you tried? These are all life. It's say, while it's easy to say lifestyle change,
It's not
I
easy
would say
to
it's
do.
one of the most difficult things to do is lifestyle change. So I would say I find lifestyle change very difficult. So it's difficult to put a pressure on somebody and say, do all this at 20, 30, so you don't get dementia when you're 60, 65. Keep your mind active. So we done research like you speak more languages, you can delay dementia by 5 years. So
Yeah, it's
all
not
of this
bad to
is
be in Karnataka where we have to learn Kannada. So
so
it's fine.
all this is nice, but I don't know, like Dr. Ravi said, once the amyloid cascade pathway sets in and the proteins start aggregating in the cells, I think that at that point you need good biology-based medication. We need therapy for these people. Yeah.
So Ravi, you want to say what we should change, which will make
I
a
mean,
huge impact?
the biggest thing when it comes to cellular level, the biggest issue is stress, cellular stress. Of course, it's connected to your overall stress. So, reducing the stress is one of the major thing, as also Professor Suvarna mentioned. Reducing stress by any means, I think, will be the biggest thing. And other major thing is the like health related things. One of the biggest risk, major risk factor is cardiovascular thing. So like, the comorbidities because of the hypertension and diabetes. So if these can be detected much earlier, if a person knows that if they have diabetes, they have hypertension, I think controlling them would be one of the major things that can, to some extent, reduce the risk. But one more thing I want to mention is like, which should happen is, so only very small percentage of dementia is caused by, at least as we know, is because of the genetic cause. The majority of that so far we call it as sporadic. But even when we call it sporadic, there are many genetic factor influences that. This we are only just beginning to understand. So we need to understand the other genetic factors which could contribute to the risk. Only one thing we know is ApoE4 allele, but which is also not very clearly studied in Indian context. But there are very likely, without doubt, there are
I
many
think
other
genetics
genetic
in
factors.
Indian context is what is missing, right? As in, we know other genetic factors in different populations, but not for Indian population, I guess, right?
So ApoE4 is a very well-established genetic risk factor, but is primarily studied to a great extent from the European ancestry.
Exactly. Yeah.
So, and it is not studied to the same extent, not even close to the same extent in the Indian answer, like, genetic ancestry. So whether ApoE4 would have the same deleterious effect in the Indian population is something, I mean, it probably does, but it's a question which needs to be clarified further. It's not clear yet, as per my understanding, but there are might be many other unique Indian risk factors. We don't have enough genetic information even to identify that.
Yeah.
I think this is one of the key sector we need to probably work on.
Correct. And Isaac.
I would say physical workouts. I think there are well-known listed risk factors for dementia, of course, from the Lancet paper, the finger study, etc. I mean, the studies of which are happening very much in CBR as we speak. But I feel like if you had asked just one, I would say working out and strength training. And I'm again speaking from practice because, my mom, she did not do strength training all her life. And I'm almost like slowly encouraging her to do strength training because the evidence is very, very persuasive. And it relates to even factors as simple as grip strength, for example. If you look at grip
That's
strength.
one of the markers people use, right?
Absolutely. I mean, somebody has asked a question on preventative medicine, like in preventative medicine, when they look at, let's say, your healthy aging markers, grip strength, VO2 max, etc. Now, it's not easy for somebody who is 60, 65, and I so agree with Dr. Suvarna Alladi that everyone knows what the main, changes need to be made, but it's very hard to make them. And I think no one can do all of them.
You can start one by one.
I feel, you can at least start with one. I would say strength training and sort of lifting weights, because that's what evolution used to be. But our bodies have just now, become so sedentary. So if to change one thing, that would be.
And attend talk by standing. I think that's a great change I'll do. So there's a question for Dr. Suvarna in the boxes. Is it that there are photoacoustic imaging which has been used for brain? Can it be used for brain aging? I don't know what is photoacoustic. Someone, Saeed, who has written that. Photoacoustic imaging for brain. Is it something which is used in the field for studying brain aging? Said, is it
ultrasound? Ultrasound? If he means ultrasound, then I can.
But Said, you can unmute and ask.
Anyway, I don't know the answer. Let's be guessing.
I have a feeling this is about terawave imaging, because I think in IISc we have a group who works with this longer wavelength and they can penetrate. I don't know if Said is still around, but so are there different kind of imaging modalities being developed? So how do you, for example, detect the brain amyloids? That's only through CSF detection, right? Or no? Is it through imaging?
No, that is imaging. So that is amyloid imaging. So you have all these radioactive tracers, flu metamol, flu beta pair, several of these amyloid tracers. So the gold standard for detecting amyloid in the brain in a person is amyloid imaging. So CSF is only second best.
I see. So Syed is here. Syed, do you want to put this question to Professor Suvarna?
No, I looked up photoacoustic. I looked up, there is something. It's ultrasound and laser. Ultrasound I can tell you more about. Focused ultrasound is being used in a remarkable ways in therapy. But getting a call. Anyway, it doesn't matter. So it is being used as a marker, as for treatment of in tremor, in stroke, all of that. But and it has effects at the cellular level, focused ultrasound. But photo seems to be laser and ultrasound. I don't know about.
I see. No, I know some group in IISc developing a new technique. It's called tera imaging, and that's a tera wavelength, which is much higher, longer wavelength. So I thought maybe Syed is saying that. Okay. So there is also a question about Ravi. So there is question which people have asked before. So are there animal studies which, for example, deals with low carb or zero sugar and looking at synapses? Does it affect? So does diet affect synapses in animal models?
Yes. There are a lot of studies on the effect of diet, particularly either by reducing carb or giving only higher protein. Many of these studies, not necessarily with the dementia, this thing, but as a in general diet factor and how it also affect the brain. So in general, low calorie seems to have a beneficial effect on overall long term. But I think it's not very direct. I think it's connected with your overall cardiovascular risk and also BMI and these factors. So, but definitely low calorie low calorie is a very well-known thing with respect to longevity. It increases longevity. So definitely it will benefit the.
That definitely benefits. So I think we can, I was just sending a note that we will wrap up in another 5 minutes. So if people have more questions. So I think there is a very general question which was interesting. It was like, what do you think about aging and natural selection? So why do we all age? Is there at least brain aging? Is there an evolutionary explanation to this, or I don't know who wants to take up this question?
Aging is clearly is not doesn't have evolutionary pressure. Because particularly once
it doesn't make any you have already reproduced, passed on your genes.
Correct. Yeah, exactly. But especially as we know that, like, dementia starts in 60 plus, when most of the humans, like, male or female, have crossed their reproductive age. There is no chance of their, like, so evolutionary pressure is not there. And that could be a contributing factor, because since the evolutionary pressure is not there, there is no reward for keeping the genes accurate. Expression of genes or keeping the DNA damage, all these things need not be taken care of, because which is very essential when you are in the reproductive age, because that pressure is lifted, so there is more relax on the DNA repair, and there is more accumulation of all kind of mutation. Clearly that's a contribution. So aging doesn't have a selection pressure. So
Correct.
that's what I would say.
Yeah, I think it's just we are in this unique opportunity in evolution where we are beyond the selection somehow, right? And
Yeah.
like we are not getting killed by diseases or by predators. So I think we are bound to age, but I think we all want to age
Yeah,
healthily, have a
I think we are
healthy,
not
graceful.
expected to live beyond 50, I think.
Yeah. So I think last few questions. Any connection with gut microbiome and brain aging? I think there's a lot of connection. Some of anyone wants to take up this question? Is CBR doing anything with gut microbiome and brain aging?
We
Ravi?
are not doing it, but I
Not.
know there is a study going on in St. John Medical College, and I'm sure there are a lot of studies going in Pune and elsewhere. So clearly there is a link.
Yeah.
So people have identified that gut microbiome particularly changes in much later age, 80 plus. So, and that they say have a impact also on the neuroinflammation, which is one of the cause of the aging.
And I was hearing a TED talk, and it says that our gut has almost equal amount of neural connection as our brain. Uh, so if you have a neuroinflammation starting in your gut, of course you will be affecting neurons also in your brain, right? I think definitely it has to have a lot of implication, gut
Yes.
microbiome on. Yeah.
Yes,
Okay.
definitely.
So I think one question about biomarkers. What is the thought about what are the new multi-omic biomarkers which are currently in the field? I think we talked about a few, tau and amyloids. What are the other new biomarkers which are currently coming up?
So among the phosphorylated tau, it's P217. Now that's the first US FDA approved biomarker.
Okay.
It's actually approved by US FDA for clinical use. There's a phosphorylated tau 181, and then that is for amyloid and for Alzheimer's pathology in the brain. And then you have for inflammation we use GFAP. See, these are the there are plenty of proteins that are abnormal. So if you
I
talk to
see.
a proteomics expert, hundreds of thousands of proteins are abnormal, and
But which
every
one will you tell in your clinic, to go and test for?
that's we won't tell so soon.
I see.
Tomorrow. No, we do not want every worried well person with mild memory problems to go to Vijaya Diagnostic and get a P217 and not know what to do with it. That is a tsunami in the making. And as clinical cognitive neurologists, that is one of the things we are really pushing back on. We are very excited it's there.
Yeah.
And so it has to be used in a person who's complaining of in the clinical setting. Research is a different thing. You take consent, you do all of that. So it's ethics approval, consent, and then you can definitely study for many biomarkers. That's different. But in clinical setting, I think today, if there are cognitive complaints, you can do a P217 clinically. Having said that, these biomarkers have been validated in Western white Caucasian populations. There is enough evidence that in America itself, that between Africans, Hispanics, non-Hispanic blacks, there's a lot of variability in the not lot, significant enough to cut to affect the cutoffs. Like random blood sugar, HbA1c above 5 or 6 is like diabetes. Like we are not at that stage yet. We need to do patient studies in our populations. There's also evidence that renal failure, glomerular filtration rate, obesity, diabetes, hypertension, stress, socioeconomic status, education, all of that affect biomarker levels. So unless we validate them in our populations, we should not be using them.
Correct. Correct.
But
So I think
they
there
are
are.
coming. But they're coming.
Yeah.
They're there now. People are using them. And we are like running very fast. We have a large validation study that we have done from across, and it's so complex that we have to put it out there, develop cutoffs, and say sensitivity, specificity, positive predictive value, negative predictive value, but they're already available. So this is the thing, like you have Coca-Cola coming in before you have drinking water. This is always the problem for us
Yeah.
clinicians. So then there is NFL, which is a good biomarker for
Mm-hmm.
neurodegeneration. So like that, there are, and it's not going to be one biomarker. It's going to be the ratios. It's going to
Yeah.
be vascular disease biomarkers. So many
Yeah.
other biomarkers in combination will help us make a diagnosis. That's the future. Yeah.
Yeah. So I think there was another question which falls in the same category. What about free DNA in the cytoplasm or mitochondrial DNA in the cytoplasm, which are all signs of inflammation, I think. But as you said, there are very few validated biomarkers which clinicians can use. And I think we need to do more studies of connecting the pathology with the biomarkers, right? And yeah, okay. Um, so is there any effect on physiotherapy currently? Okay, I think there was one question about diabetes and dementia. So, are there big studies in India, Ravi or Suvarna, where we are correlating diabetes and dementia in India? Or
So
there are already data
there,
sets?
no, there is some data, and we're also doing one large NIH study. There's a diabetes foundation, Mohan Diabetes Foundation. They have a large cohort of diabetics. We'll be doing all these biomarkers and imaging. Even otherwise, there are enough associative studies. The anti-diabetic drugs, Ozempic is a
Yeah.
big drug which is being,
Mm-hmm.
probably will be a game changer in dementia prevention also. We don't know. So Dr. Ravi could probably tell us at the neurobiological level what he
Yeah.
thinks.
Yeah, I mean, this is a very interesting area. One of my PhD student is actually working on exactly this. But working on this animal model and cellular model is even very, very tough. How do you create a diabetic condition at the cellular level is a major challenge.
Yeah.
But I can tell that there are substantial amount of like studies which are going on. There is a clear link which is even shown in the human studies. I think even Lacidats had a component on that, and there is a link is established. But cellular and animal studies are going on. Uh, but there are huge challenges with respect to the what exactly the right model to study this. That's something we are working on that.
Yeah. Yeah. So I think I'll do a last question, which is about brain activity. Like for example, when we are all employed and we are doing a lot of brain activity, we have a job and so on, versus when I'm retired, right? When I probably my brain activity, as in if I have not found enough engagements, is there a effect on dementia or on cognitive scores when people are more free than when they have less engagements than so your brain is not getting so much stimulation. So Isaac, any thoughts on that?
Anecdotally, at least I can share that, I think that correlation is true. We don't know whether
Hmm.
it's causation, but that
Hmm.
correlation of, somebody having, it's almost like the brain makes up for, your shortfalls while,
Hmm.
you're actively engaged with something, right? And when that purpose goes out, you will suddenly see like a dip. We work with, I would say 20% of our, clients on a month-on-month basis are actually above the age of 60. And anecdotally, I can tell you that this is true for a lot of people, wherein either kids or themselves, they'll come and tell us that till last month, he or she was doing absolutely okay.
That's what
Retirement.
I also feel. So because I've seen my parents when
Yeah.
when yeah, they had less engagement, they were probably less happier and
And it's like, it's
yeah.
not even like gradual decline. It's almost like
Mhm.
a, very steep decline. And one of
Yeah.
the more realistic, at least like references in popular culture is this film called Still Alice, which is based, by the way, directed by a person who himself had ALS. He passed away.
Mm-hmm.
So extremely sensitive portrayal of a professor in a university who
Mm.
was doing fine, retires, and then suddenly. So I think that reference always maybe has made me biased towards thinking so, but anecdotally for sure, I would say that there is a correlation.
Yeah. No, I think it was. I had a lot of fun in this discussion, and I personally got to know all of you. This was great. And okay, there is still something people are typing, but I think we are almost closing. And I think what I got from all of you is that to prevent, we have to have physical activity, look for diet changes, also mental activity, right? And there are, I think, interesting research coming in the field to detect biomarkers and so on. But it will take some time. But I think in coming years, we'll look forward to these new therapy, new biomarkers, and cohort studies for Indian population. And yeah, everyone who can be part of such studies can contribute to this research, right? And yeah, so great. I think we will close the webinar now, and thanks everyone for joining again. Yeah.
Thank you
See
for
you.
having.
Thank
Thank you
you.
for a
Bye-bye.
wonderful
Thank you.
great
Thank
Thanks,
you very
Suvarna.
much,
Thanks,
everyone.
Ravi. Thanks, Isaac. Yeah. Bye-bye.